First of all, thank you for the invitation to be presenting here. My name is Thiago. I'm from the University of Texas at San Antonio on the Department of Chemistry. I'm from Brazil. I finished my PhD in the University of São Paulo, and now I'm doing my postdoc here in the UTSA. So my presentation today is going to be about halotolerant and halophilic enzymes and their applications in the reducing agroindustrial residues and avoiding water wasting. So talking about first agroindustrial importance, you can go on with the slide, please. So as we can see here, we have different pictures from the World War II, where all the countries started to invite people to grow your own food, to produce your own food, because they were having not so much to provide for the people. So all the world started to improve production and distribution of food, start to raise the interest of improving the production and distribution of food. Next, please. And then was created the Food and Agriculture Organization by the UN, the United Nations. So it was created in 1945 to restore the food system and solve hunger and malnutrition problems. And nowadays, the agriculture has become a major resource to reduce poverty and contribute to improving the standard of living of world's population. Next, please. So as we can see here, we can see that the most polluters of the world are China, India, United States, and Brazil. And they are also the major food producers in the world. So they are using these residues from the food production to be burned because they don't have nothing to do with that. So as we can see here, China, India, US, and Brazil are generating a lot of residues from food production. So this is a problem. We started to produce a lot of food, but we don't have alternatives to these residues. And also, as we can see here, you can go on with the slide, please. Yeah, one more, to the end of the slide. Yeah, that's it. So as we can see here, the same countries that share the most pollution in the world, they are also the most water wasters in the world. So this is not an unfortunate coincidence. As we can see here, China, China, US, Brazil, and Russia, they're also wasting a lot of water and producing a lot of food, residues from food. You can go to the next slide, please. So this is not an unfortunate coincidence. As we can see here, globally, 70% of the fresh water is used for agriculture. So the most agricultural producers in the world are the same that uses more water to produce it. You can go to next slide. So we have new problems now. So we're producing a lot of food. We have food for the population, but we are generating a lot of residues and we are wasting a lot of water to produce food. How to deal with it? How can we manage this? Go on, please. So the agro-industrial residues, they can be turned from the trash into cash. How? The waste management, including the reuse, recycling, and valorization of wastes and byproducts, has recently become a cost-effective solution to transform high volumes of low-cost materials into high-value products, improving the circular economy. You can click one more, please. Yeah. So we have the agro-industrial residues that can be managed by reducing, reusing, recycling, improving the circular economy. So go on, please. Next slide. Inside the circular economy, the agro-industrial biomass represents one of the most promising reserves for production of sugar, renewable energy, biopolymers, and bioplastics. So we have different kinds of agro-industrial waste. For example, here I used some of these pictures. As you can see, I use a rice straw, I use a paper sludge, I use eucalyptus chips. So we can use all these residues to produce high value added compounds, as for example, bioenergy, biofuels, biopolymers, and bioplastics. Next slide, please. The agroindustrial waste in general, they are composed by lignin cellulosic materials, which comprises cellulose, hemicellulose, and extractives. And the cellulose is a linear homopolysaccharide consisting of beta-glucose. As we can see here, is a structure pretty much simple of only beta-glucose linked by 1,4 linkage. Okay, so it's a homopolysaccharide sugar. Go on, please. Next slide. And on the other hand, we have the hemicellulose, which is a heterogeneous polymer composed of many sugars, such as xylose, arabinose, mannose, glucose, and galactose. So we have C5 and C6 sugars. So it's a very complex structure. The hemicellulose has all these structures inside its complexity. Go on, please. Next slide. And it is also known as the second most abundant carbohydrate material in the nature, consisting of 25 to 35 dry weight of agricultural materials. So the hemicellulose is the second most abundant residue in the agroindustrial residue. So the major hemicellulose component of agroindustrial residues is the xylane, as we can see here, the structure, which is a polysaccharide composed of a backbone chain of chylopyranose units linked by beta-1,4 bonds. So the main chain of the hemicellulose is the xylane. Okay, keep this in mind. The xylane is the main chain of the hemicellulose. Go on, please. Next slide. And now let's talk about the xylanases. These are the most common biotechnological used enzymes. Next, please. And the production of these enzymes and the environment-friendly conversion of the residues to other bioproducts can represent current and viable alternatives for the waste generated by agro-industries. So we have the xylanase and we have the xylane, the majority of the hemicellulose residues. Next, please. The xylanases can be used on the hemicellulose residues, for example, biomass, to produce a lot of different sugars. So we can use xylanase to produce xylooligosaccharides, different sugars, as for example, xylose, xylobiose, xylotriose, tetrose, pentose, hexose, and moving on. So the majority of the products generated by the xylane from the xylanase activity are the xylobiose and xylotriose. So using the xylanases on the agroindustrial wastes can generate a lot of xylooligosaccharides. And the xylooligosaccharides are so much important for the industry, and they can be much more valorized than they are today. They can be used as antioxidants, antimicrobial drugs, anti-allergic drugs, anti-inflammatory drugs, prebiotics, and sweeteners for diabetics. As for example, the xylitol, which is the most known sugar for diabetics. Okay, I'm talking about xylanases, about agroindustrial residues, biomass. But what about the water? Where am I going to save the water? Go on, please. So here's the trick. We can use halophilic or halotolerant enzymes on the agroindustrial recycling. What are halophilic and halotolerant enzymes? These kinds of enzymes are resistant to high salt concentrations, so they can be active under high concentrations of different salt. So here's the idea. We can use halophilic, halotolerant enzymes with seawater instead of using potable water, and then we can do the enzymatic hydrolysis of these residues generated from the food processing and food production, And then we will be recycling these agroindustrial residues to more value-added compounds, such as building blocks or biofuels or halophilic. And also, we are not using potable water to do this hydrolysis. We are using seawater, so we are saving water. And more than that, we are not just saving water, but we are increasing the ability to reuse the products that wasted a lot of water before. So we are increasing the yield of the production from the water that was wasted from the food production. Next, please. So the halophilic and halotolerant enzymes, they came from microorganisms that in general inhabit extreme environments as saline sources. Next, please. And these enzymes are defined as biocatalysts with the ability to catalyze reactions in the presence of high levels of salt. So they have high salinity tolerance. Next, please. And one of the key factors for this kind of enzymes can be the Aspergillus species, which are enzymatic machines and novel sources of evolution. The Aspergillus species, they are historically efficient biomass decomposers, and they are worldwide distributed. Next, please. So therefore, this fungi retain enormous genetic evolutionary potential to be tested and discovered, because this fungi can be found in every corner of the world. So they have a lot of different genetic and evolutionary information that are sometimes hiding inside their structure. And especially the Aspergillus clavatus, which was used in this work, it shares 98% sequence similarity with Aspergillus fumigarus. And the Aspergillus fumigarus is a well-known species, common found in arid environments, high temperatures, and consistently appears in solar Saturn. So Aspergillus fumigarus has already been described as an halotolerant fungi, and also its enzymes. Next, please. And Aspergillus clavatus is a much less studied fungus and has only few enzymes already described in the literature. So Aspergillus fumigatus is a well-known species. We have a lot of different literature information about this fungi. And Aspergillus clavatus is not well-known species. And also, Aspergillus clavatus is similar to Fumigarus. So the idea is Aspergillus clavatus could have some interesting information for enzymes, halotolerant or halophilic enzymes. Thinking about that, we started a screening of ligulocellulosic enzymes in different residues, and we started the culture standardization of the endo beta-1,4 xylanase. Okay, so we used different residues for this work. We used corn cob, bean straw, corn husk, barley bagasse, rice straw, eucalyptus chips, coffee husk, and paper sludge. And the paper sludge was the best residue to produce the endoxylanase by Aspergillus clavarus. And then we started a production of 33 units per mil of enzyme during the screening. And the next step was the standardization of the culture of this fungi, and we reached 192 units per mil of enzyme production after the pH standardization. So we used different culture conditions for the production of this enzyme. We did pH standardization, we did temperature standardization, and next please, and we changed the concentration of paper sludge during the culture, and we finished with a standard culture condition. We can go to the next, please. We finished with a standard culture condition with a high xylanase production. You can go to the next, please. So then after standardization of the culture production of the endoxylanase, we started purification of this enzyme, because this the fungi enzymes, they are in general produced as extracellular extracts. So they are extracted from the media, because the fungi has this capacity to send out of the cell the enzyme. So we got the enzyme from the media, and then we started purification. We used biogel P60, which is a size exclusion chromatography, and we got at the final purification 1 milligrams per milliliter of xylanase and 7 mils with an activity of 373 units per mil. Here in black we have the activities, and in blue we have the protein absorbance. So as we can see here in the coma C gel, we have the protein pretty much pure, and also we did a silver staining, and we got the pure protein as well. The molecular mass of the protein was estimated as 25 kDa. Next, please. After the purification and verifying that the protein was pure, we started the identification of the amino acids of the protein, and we got some sequence here. So we cut the band of that gel, stained it with Comasi, and sent to mass spectrometry, and then we got 100% of identity with an endoxylanase GH11 from Aspergillus clavatus, which was predicted by Segato et al. 2014. So this enzyme has been predicted to exist before, but this is the first time that we saw this enzyme really in the extracellular production. Also, we did the typical structure, we did the three-dimensional structure prediction of this enzyme. As we can see here, it has a typical structure of glycosyl hydrolases 11 with a jelly roll conformation and with no carbohydrate binding modulum 1 type, which is common for glycosyl hydrolases from family 11. And we did the phylogenetic relationship of this xylanase from Aspergillus clavatus, and it was shown that this enzyme has different properties when compared to the other xylanases from other Aspergillus species. So it's a pretty much uncommon enzyme, as we can see here through the phylogenetic relationship. It shares around 7% of similarity with other xylanases, so it's not so much. Next, please. So this was the first report of obtaining this enzyme in the extracellular extracts from Aspergillus clavatus. Next. After that, we estimated the optimum pH, and what it was 6, as we can see here. We estimated the optimum temperature, as we can see here, was 55 degrees. And then we did the stability at different pH and the stability at different temperatures. As we can see here, the enzyme was pretty much stable after 24 hours in pHs ranging from 3 to 8. So it's a pretty much stable enzyme. And also the enzyme kept around 80% of its activity after 1 hour in 50 degrees. So it's a good enzyme with good thermostability as well. These results are in accordance with many purified xylanases from the literature, where we can find xylanases with optimum pH around 5 and 7 and temperatures between 40 and 60 degrees. Next, please. And then we started to evaluate the effect of sodium chloride on the xylanase activity and stability. So as we can see here, with the concentration ranging from 0 to 3 molar, we left the enzyme in the presence of sodium chloride for 24 hours, and then we evaluated the activity. Here we have in 100 the standard reaction with no sodium chloride. Okay, so we have 100% of relate activity, and then with sodium chloride in black. So as we can see here, with 0.5 millimolar 0.5 molar of sodium chloride, we had an increase of the activity, and then up to 3 molar of sodium chloride, we have more than 85% of enzymatic activity. So we used the stability here after 24 hours, and then we made it the reaction with these same concentrations, okay? So we use the stability and also the reaction. And we have the stability and the reaction pretty good for this enzyme. So according to Carly et al. 2016, enzymes in the presence of ions may associate to them, stabilizing its structure and consequently improving its function. In addition, salt tolerance could be associated with alteration in the molecular surface, with a lower content of bulk amino acid residues and higher content of charged, polar, and small amino acids. This combination allows the hydration of the enzyme at high saline conditions. So you can go next, please. And as we can see here, this is the amino acid composition of this enzyme. Around 56% of the enzyme structure is composed by charged, polar, and small amino acids. So this could be an explanation of why this enzyme likes and is still stable after 3 molar of sodium chloride. Next, please. Then, at the end of the work, we did hydrolysis. So keep in mind that we did two different processes here. First, the production using paper sludge for carbon sources for the fungi. And the second step was to use the enzyme on the paper sludge to generate sugars from this hydrolysis, because paper sludge is composed by hemicellulose and cellulose. And here we are breaking the hemicellulose. So we did this hydrolysis using no sodium chloride, and we got around 2.6 micromolar per milliliter of reducing sugars, mainly xylose, xylotriose, xylobiose. And using 0.5 molar of sodium chloride, we got almost a double of reducing sugars, reaching 4.5 micromolar per milliliter of reducing sugars. Next, please. Yeah. So as a conclusion, we can see that using the agroindustrial waste and also using not potable water can be healthy for the environment, decreasing the avoiding, of course, the global warming, because we are reducing the accumulation of biomass. We are turning in more interesting the process of recycling these agroindustrial residues, and we are not wasting more water to do that. So we are optimizing the production of products from the recycling, and also optimizing the use of this first water for the production of the food. And also we are generating money, of course, for the industries. We are generating more products for the population, as for example, prebiotics, medicine, and health for the people. And also we can use a sustainable way to do that, because we are recycling, we are avoiding to use more water to produce more products for social interesting. So with that, the endoxylanase present a good pH stability and tolerance over wide salt concentrations, and these observations make this enzyme interesting to be applied in enzymatic cocktails for biomass hydrolysis with seawater, because seawater has around 0.5 molar sodium chloride, which would be contributing to improve the economic viability of oligosaccharides, second generation ethanol, and other biochemicals production. Next, please. So here are my publications for this work. The first one is about the screening. The second one is about the standardization of the culture conditions. And the last one, next please, is about the purification and the application of the enzyme on the hydrolysis of the paper sludge. Next. Oh, yeah. So my acknowledgement, this work was developed on my PhD at the University of São Paulo in Ribeirão Preto. My advisor is the Professor Dr. Maria de Lourdes Teixeira de Moraes Polizelli, and these are also my contributors for this work. CAPS was my funding agents from my PhD. And next. And this is my current home. I'm at the University of Texas at San Antonio. My current advisor, Dr. Alder Lamb, and my lab mates here in the right. Next, please. That's it. Thank you.